Okay, so that's an invitation to think about fermentation and what the possible limitations are. Okay, so what do you think about this? Okay, this is what the votes are. So end products can be toxic. Yeah, that's true, that's correct. Energy yield per carbon is low. A low yield of ATP per glucose molecule. So definitely a limitation. Requires oxygen. This is wrong. Okay, number two is the wrong answer. Never forget fermentation occurs without oxygen. It's anaerobic. And it doesn't require another electron acceptor because the electrons are going to be pushed onto pyruvate. The pyruvate comes from the glucose, so there's no external electron acceptor in the process. You just need access to the glucose at the beginning. That's it. Okay, so fermentation, I think, is kind of like the simplest situation, and the rest of what we need to do is the more complex stuff. So, summary of fermentation. Okay, the problem is that glycolysis produces too much NADH relative to ATP. The electron acceptors are all derived from pyruvate somehow. Okay, the end product of glycolysis. It's useful for us because, you know, cheese, yogurt, this kind of stuff, they're all food products produced by bacterial fermentation. And it will only occur in the absence of oxygen or when there is a lot of sugar. So that's important for understanding what happens in the TP, in the lab classes, because, for example, if you remember last lecture, I talked about Chapman agar, which is differential for Staphylococcus aureus because there's a large concentration of mannitol in the agar. And I said, yeah, the fermentation of mannitol acidifies the medium and you get the color change. But if fermentation only occurs without oxygen, how can this happen? Because the bacteria are growing on the agar plate. Well, it's because there's so much sugar available that the bacteria, they don't bother doing anything else. They don't bother using respiration. They'll just ferment it because the pathway is very simple. Okay, so if you've got a lot of sugar around, bacteria will preferentially use fermentation as a means of getting biochemical energy, even if oxygen is present. Okay. Right, so now we're going to talk about respiration. So in this case, there's basically three stages. So glycolysis, so you end up with pyruvate, and then the Krebs cycle, which is this, which I'm sure you'll be doing in biochemistry. So you get pyruvate to acetyl coenzyme A, which combines with oxaloacetate to give you citrate. So 2 carbons, 4 carbons, 6 carbons here. And then the 2 carbons that come from the acetyl coenzyme A are progressively oxidized and kicked out as carbon dioxide. And since they're being oxidized, they lose electrons, right? And those electrons give you more NADH. So at the end, the bacterium has still got to get rid of all this excess NADH, and it does so by transferring them down an electron transport chain which ends up with oxygen. So you get oxygen reduced to water. And oxidative phosphorylation allows mitochondria and bacteria to produce ATP from this process. So we're not going to be worried about the Krebs cycle here, okay? You just have to realize it produces NADH. So we'll pick it up from there. So in E. coli, we're starting off from here, NADH, and the first thing that's going to happen is the electrons are going to be transferred by this enzyme, the NADH dehydrogenase, and they'll be transferred onto these flavine adenine dinucleotide iron sulfur complex, and then to coenzyme Q. Coenzyme Q is a very interesting molecule here because it's got this kind of like isoprenyl bit that is soluble in lipids. Well, the whole thing is soluble in lipids. And then you have this cyclic structure here. So this is the oxidized form. And it can very easily be reduced to give you 2 hydroxyl groups here and an aromatic ring here. So that's the reduced form. So the enzymes, the electrons come from NADH, get transferred onto coenzyme Q, which then is in its reduced form, QH2, and then it will interact with a cytochrome oxidase complex, which is going to transfer the electrons to cytochromes in here, and then they'll get shuttled down and then finally transferred onto oxygen to give you water. Now the clever thing about coenzyme Q is that it will pick up the protons that are on the cytoplasmic face of the plasma membrane, and then when it is reoxidized, when it gives up those electrons here, it will release the protons on the periplasmic face of the membrane. So just by doing that, it creates a proton gradient. Now, depending on how much oxygen is present, E. coli will either use the BD complex or the BO complex. So the BD complex is expressed when there's not very much oxygen available. So this has a high affinity for oxygen, but it doesn't actively transport protons. Whereas the BO complex has a low affinity for oxygen, but it's useful because as well as transferring the electrons that come from coenzyme Q onto oxygen, it will also export a couple of more protons, so it's better for producing a proton motive force. So cytochromes, what are they? Cytochromes are these small proteins with a heme group here, and they are essential for transferring the electrons from coenzyme Q onto molecular oxygen here. And they all contain iron. So as I said, iron, it's an essential growth factor for bacteria because you have a lot of iron here and you have iron in the cytochromes, which are necessary for this electron transport. So this step generates proton motive force from NADH. And finally, the proton motive force is used to produce ATP with the proton ATPase. So 3 protons are going to be transferred across from the periplasm to the cytoplasm, and this allows the generation of 1 molecule of ATP. Now, this is really great because if you work out all the calculations, apparently the maximum yield is about 38 ATP per glucose molecule, compared to 2 ATP per glucose just for glycolysis. So it's almost 20 times more efficient in terms of energy extraction. Okay, so that's aerobic respiration. Now, some bacteria can also perform anaerobic respiration. Now, just think about that for 1 second. Anaerobic respiration. This shouldn't exist, right? What is that even supposed to mean, this word? Well, what it means is that some other electron acceptor is being used instead of molecular oxygen. So Paracoccus denitrificans, for example, has got an electron transport chain kind of like this. So it starts out the same. You've got the NADH dehydrogenase, coenzyme Q cycle, passes the electrons onto some kind of cytochrome enzyme, but instead of passing the electrons onto molecular oxygen, it's going to pass them onto nitrate, which will be reduced to nitrite. And it has other cytochrome proteins in the membrane which can pass electrons onto nitrite and then end up reducing the nitrite all the way to dinitrogen gas. So why does this work? Well, this works because NADH is a very good electron donor and nitrate is a very good electron acceptor. So nitrate to nitrite, very favorable. Nitrate to dinitrogen gas, very favorable. So as long as nitrate is available, then, bacteria can perform anaerobic respiration and extract a lot more ATP per molecule of glucose than fermentation. So that's why they use it, okay, because the ATP yield is so much better. And in fact, nitrate is not the only possible electron acceptor. So other bacteria can use sulfate, iron or ferric iron, arsenate, selenate, or I don't even know how to pronounce this, selenate, if they are present. And in an anaerobic environment, these different electron acceptors will be used in a kind of strict order. So the best electron acceptor, which is molecular oxygen, will be used first. So this is, bacteria close to the surface, on the ground, where oxygen is available. Then there'll be aerobes, and then a little bit further down in the water table, you'll have bacteria using nitrate because it's the next best electron acceptor. When all the nitrate is used up, you'll have bacteria using manganese, then iron, then sulfate, and then finally, when there's nothing left at all, apart from maybe a little bit of carbon dioxide, then methanogens can use carbon dioxide as an electron acceptor and produce methane. All of these electrons come from where? They come from the organic carbon at the beginning of the process, because we're still talking about chemautotrophs here. So all of these inorganic compounds can be used as electron acceptors because it's still energetically favorable to transfer electrons from basically glucose to begin with down to these other electron acceptors. Now, the one that is a little bit weird here is that how this can possibly ever work. Okay, but it does. So often methanogens will actually be using molecular hydrogen as the source of electrons. Okay, so yeah, I'm going to ask you another question now, so to see if anybody has understood what is happening during anaerobic respiration, because the concept is a bit difficult. Start, okay. Hey, not bad, not bad. That's good. Okay, interesting. Okay, interesting. So here are the votes. Okay, let's try and figure out what they are. Yeah, okay, right. So let's look what they are. Oh, I didn't know I could do that. Yeah. Okay, so the correct answer is C. Okay, glycolysis, then the Krebs cycle. Krebs cycle gives you NADH, so you transfer the electrons from NADH to nitrate. That allows you to generate proton motive force, and that gives you ATP. So that's the correct answer. 4, I kind of understand why people did this, because it's the same, except that you say that the electrons are going from pyruvate to the electron acceptor, but that's skipping out the step. Or it will go through the Krebs cycle. So answer 4 is wrong, because F is just a kind of, it's not direct like that, okay? Goes through the Krebs cycle. And C, E, A, D, and A, B, C, D. A, B, C, D, E, I just, okay, A, B, C, D, E, give me that one. So I didn't even think about that. C, E, A, D is interesting though. It's kind of, I think it's the wrong way round, right? So glycolysis to produce pyruvate, ATP hydrolysis, and then generate the proton motive force. That's the wrong way round. A comes before E. You generate the proton motive force and you use it to make ATP in respiration. Aerobic or anaerobic respiration, doesn't matter. Proton motive force first, then ATP. Okay, good. So I think everybody understands what's going on. So now we can move on to chemolithotrophs and phototrophs. How long we got? Good. Got about an hour for that, so that's good. So chemolithotrophs. There are 3 different types here: hydrogen oxidizers, nitrifying bacteria, and sulfur oxidizers. And what they are doing is generating metabolic energy from a redox reaction between 2 inorganic one electron donor, 1 electron acceptor, but they're both inorganic. There's no organic carbon involved. And what is going to happen, or the types of metabolism that can work, is you always have to use an electron transfer from an electron donor that is a better donor than the acceptor. So this will be energetically favorable. The other way around, it's never going to work. And interestingly enough, okay, for the nitrifying bacteria and the sulfur oxidizers, this is kind of doing the opposite thing to what we just saw in anaerobic respiration. So in anaerobic respiration, nitrate, this half reaction here, this was the electron acceptor, right? Whereas in a chemolithoautotroph that is going to generate energy from nitrogen oxidation, there are some bacteria that can live just by using nitrite as the electron donor and molecular oxygen as the electron acceptor. And this can work because molecular oxygen is such a good electron acceptor. Even something pathetic like this, nitrite to nitrate, is like nothing. I don't know if you could even run a digital watch on this reaction here. It's like pathetic, but it's enough to get some metabolic energy and to live and grow for these bacteria, just because oxygen is such a great electron acceptor. So let's have a look at the different types of chemolithotrophs, what their electron acceptors are, what their electron donors are. And really the question is also, how do they get NADH, proton motive force, and ATP from these reactions? So the simplest is perhaps what you have in hydrogen oxidizers. So for Aquifex, hydrogenophaga, they are using molecular hydrogen gas as the electron donor, oxygen as the electron acceptor. This is massively favorable. You know, this is like a hugely, so hydrogen is up here, oxygen is down there. You know, this will explode, of course. So, you know, it's hugely favorable reaction. And in fact, hydrogen is such a good electron donor, it's even better than NADH. So NADH can be produced directly from hydrogen gas. Okay, so that's easy. And once you've got that, then you can produce a proton motive force, and then you can produce ATP, because then you can transfer the electrons from NADH all the way down the electron transfer chain to oxygen, and that will produce ATP. Okay, easy. So you get the three things you need directly. Now it's very much more difficult for methanogens. Okay, for example, Methanococcus blue, it's an archaebacterium. So archaebacteria are methanogens, not eubacteria. Now, I'm not even going to try and understand the process of electron transfers in archaebacteria from hydrogen to CO2. It's very complex, and it's complex because it's very difficult, because CO2 is a terrible electron acceptor, right? It's the worst, But it's still possible, okay? And these bacteria produce methane. Okay, I think if I kind of remember correctly, there's a kind of, no, forget, no, I'd better not go into it, because A, it will take time, and B, it will probably be wrong. So, nitrifying bacteria. Okay, so you've got some kind of reduced form of nitrogen as the electron donor, oxygen as the electron acceptor. Now, Nitrobacter is the simplest one, because it's going to just perform the oxidation of nitrite to nitrate. Nitrosamonas, ammonium to nitrite. I don't know, I always get mixed up with these two, which one is doing what. So nitrosamonas sounds like ammonium, right? Okay, so maybe that will help. They use ammonium, and then the Nitrobacter gets a nitrite back to nitrate. Anyway, that's what I think anyway. So one of the problems here is that this half reaction is, you know, nitrate is a very good electron acceptor. Nitrite is a lousy electron donor. So there's no way, if you put these two half reactions together, it's never going to go in that direction. We saw that. Anaerobic respiration is running the other way around. You've got NADH, and the electrons go on to the nitrate. So how can these bacteria get it to work the other way? That's difficult. It's not easy at all. NADH can't be produced directly. So the key enzyme here is the, I think it's called the nitrite oxidoreductase, which is in the plasma membrane of Nitrobacter, for example. So it's performing this oxidation reaction on the plasma membrane, and somehow it manages to transport one proton across the membrane as it's doing this. So this reaction is producing proton motive force, and then that can be used to produce ATP with the proton ATPase, and it can also be used to push electrons onto NADH. How can that work? So if you remember when we talked about the proton motive force and the Nernst equation, you can build up quite a big potential difference if you've got a high gradient. So that means the proton motive force can be enough to push this reaction in the other direction. Okay, so that's reverse electron transport. But you'll need to, oxidize quite a few molecules of nitrite to be able to get enough proton motive force just to generate one molecule of NADH. It's not like one for one, okay? So that's how these bacteria can actually manage to do this. Only possible in the presence of oxygen, because oxygen is the only electron acceptor that's good enough. Okay, sulfur oxidizers, similar kind of thing. Okay, so you have reduced forms of sulfur, which are the electron donors. The electron acceptors are either molecular oxygen or nitrate. Okay, so this works because nitrate is a better electron acceptor than sulfate. Okay, so how can they get ATP and NADH from these reactions? Well, the sulfite to sulfate reaction, together with, there's a sulfite oxidase reductase in the plasma membrane. It's doing the same thing as for nitrate to nitrate in the nitrogen oxidizers. So this can produce a proton gradient. And some sulfur bacteria can also produce ATP directly in the cytoplasm by this set of reactions. So basically sulfite can be transferred onto adenosine monophosphate to give you this APS molecule. So I guess it's adenosine phosphosulfite. And then the sulfite groups can be sulfate groups, because they've given up their electrons. So the sulfate groups can then be swapped with phosphate groups to give you ADP. And then this way you get ATP. So that's quite neat. Okay, you can see that, okay, sulfuric acid is one of the main reaction products here. So these bacteria are going to produce a lot of acid as their waste product. And this is a big problem in water runoff from old mines, old mine workings that have been mined for iron. Often you'll have iron and sulfide or molecular sulfur in there. Thiopilicious ferrooxidans can start growing inside the mines and produce tons and tons of sulfuric acid. Now the way to prevent this from happening is just to block the access route to the mines, because that will cut off the oxygen, so their metabolism will not function anymore. So this is a serious problem for acid runoff from old mine workings. Okay, so that's hydrogen oxidizers, sulfur oxidizers, nitrogen oxidizers. So just for all of this stuff, what can actually work as a possible type of metabolism? Okay. Oh yeah, I've got 30 answers. Okay, cool. Everybody okay for this? Yeah. Okay, so let's have a look. What do you think is possible? So glucose as an electron donor, nitrate as an electron acceptor. Yeah, that's anaerobic respiration. That works. Glucose as an electron donor, CO2 as an electron acceptor. I would say no, that's never going to work. Terrible electron acceptor. And this is basically the wrong way round. Oxygen is the electron acceptor and it produces CO2. Maybe you might think, okay, methanogens can do this, but they're really using hydrogen as the electron. Yeah, maybe methanogens. So this is basically no, not possible in brackets except for methanogens. This one down here, molecular hydrogen as electron donor, nitrate as the electron acceptor. Yes, definitely true. This one, number 3, molecular sulfur as electron donor and the electron acceptor. Apparently, yes. That is a thing that actually exists. It's called disproportionation. So some of the sulfur will be reduced to sulfide and some will be oxidized to sulfate or sulfuric acid. So apparently that can actually work and be energetically favorable. And one of the reasons is, okay, it's kind of old story about this, is that yeah, back in the ancient Earth, it was a reducing environment. There was a lot of iron, okay? So there was ferrous iron, and it would precipitate out with the sulfide. So thermodynamically, it could work because one of the reaction products is always being taken away. It's always being removed. And then finally, this one, ammonium as electron donor, sulfate as an electron acceptor. This can't work. It's the wrong way round because, why is that? Because basically you've got a hierarchy of who's a better electron donor, who's a better electron acceptor. So reduced sulfur, hydrogen sulfide, is a better electron donor than reduced nitrogen ammonium. Okay. Now don't ask me why at all, because this really is a physical chemistry question, but that's just the way it is. Okay. So sulfur oxidizers could use sulfide as the electron donor and nitrate as the electron acceptor. That will work. Thermodynamically, it's favorable. This one can't work. It's the wrong way round. Because ammonium compound, nitrogen compounds, they're basically better electron acceptors than sulfur compounds. So this one cannot work. And you have to look at the scale with the half reactions and the redox potential of the half reactions to see that. Okay, so we have about 40 minutes to think about some of these large-scale transformations, carbon cycle and nitrogen cycle a little bit before thinking about photosynthesis. So basically everybody knows that we've got the carbon cycle, so we have an equilibrium between carbon dioxide, which is being produced by respiration, either anaerobic or aerobic, and then fixation of CO2 by photosynthesis or also by autotrophs. So the amount of CO2 that can be fixed by chemolithotrophs is very, very small, compared to what can be fixed by photosynthesis. So it's almost like 10,000 times less. Okay? And in terms of photosynthesis, then about half of it is down to plants and half of it is down to marine microorganisms. Not all of them bacteria. This is including diatoms, cyanobacteria, everything, okay? Microscopic algae. Now, that's basically what everybody knows. What I want to talk a little bit about is different kinds of organic matter, how they can be degraded by microbes, not just sugar, okay? Different types of organic carbon. And then a little bit about this process, the methanogens, and what their contribution might be to global warming. So if you look at different types of organic carbon down here, up to now, we've really just talked about glucose as being the carbon source for E. coli. You put it in the medium, something like that. But in fact, free glucose doesn't really exist a lot of the time in a natural environment. It will be present as, you know, polymer, starch, cellulose, whatever. And so just talking about cellulose for now. So starch, of course, you know, we can digest it, we can degrade it down into maltose and then import that and then use it as glucose for all of our energy needs. Cellulose, of course, we cannot digest because we don't have enzymes capable of breaking this beta-1,4 glycosidic link. So many bacteria, like a ruminococcus, can do this. So these are commensal bacteria that live in herbivores. Okay, ruminants and other herbivorous animals have got ruminococcus and other bacteria that can break down cellulose. They ferment the glucose in the guts of the animals, and then the fermentation products, there'll be lactose, butyrate, maybe ethanol, whatever, are absorbed by the animal's intestines. Okay, ruminococcus. Actually, I need to go check out my notes on this because, yeah, from about here, yeah, I have a whole bunch of stuff written down here. Yeah. Okay, so cellulose, that's one thing that's difficult to digest. Lignin, so in the woody part of trees, basically only filamentous fungi can degrade lignin. There are maybe some couple of bacteria that can do this, but most of the degradation is by filamentous fungi, and all of the degradation pathways require oxygen. So, you know, that's why when wood is buried, then that's it, that's a permanent carbon sink, because there are no anaerobic digestion pathways for lignin. And as far as I am concerned, this is like basically the only realistic form of carbon storage and taking out the CO2 from the atmosphere and putting it into a permanently stored form is like through lignin. And hydrocarbons, okay, big problem, of course. These are not natural chemicals. We're talking about plastics, petrol, this kind of stuff. And there are a whole bunch of bacteria that can actually degrade some different types of plastic. There are most of the degradation pathways require oxygen. But there are some, I hate it when it does that, okay? But there are some which are able to use anaerobic pathways to degrade some types of synthetic hydrocarbons. So, you know, there is active research in analyzing the enzymes from bacteria that can degrade plastics and to try and figure out if we can actually use them to get rid of plastic pollution. One of the perhaps problems with this idea is that if we were to do this and suddenly a lot of marine bacteria could use plastic as a source of carbon, then they would suck up all the oxygen in the oceans, and then you could cause an equally dangerous environmental problem because currently all the plastic that's in the oceans is not available as a kind of carbon source for life, right? But if it's suddenly being used for respiration, where's all the oxygen going to come from? So this is kind of a risk here. If we try and get rid of all the plastic pollution by genetically engineering bacteria and releasing them, there could be environmental impacts that we won't really be able to control. Okay, so the last thing is to talk about methane here. So methanogens. So methanogens are able to I know what the problem is. The problem is if I do this. Yeah, so methanogens are able to perform this reaction. So they use molecular hydrogen as the electron donor and carbon dioxide as the electron acceptor and produce methane. And this is just about thermodynamically favorable. Okay, if you look at the redox potentials here. So molecular hydrogen is such a good electron donor, it can even use carbon dioxide as the electron acceptor. So where do you find methanogens? Well, where you have anoxic environments that are rich in organic carbon, because some of this organic carbon is going to produce hydrogen gas as a fermentation product by other bacteria. So you find that in ruminant guts, marshes, and also coastal oceanic sediments. Now this is kind of important for global warming because methane is a greenhouse gas 25 to 30 times more potent than CO2. Than CO2. So this is why raising a lot of animals to eat them for meat is not environmentally neutral, because all of the methanococcus in their guts are producing methane, and it's actually got a significant impact on greenhouse gas emissions. The other big worry here is that in these oceanic sediments, the methane that is produced by the methanogens is frozen in complexes with water in what are called methane clathrates. So these are currently just like frozen under the water. But if the Arctic really begins to heat up, the risk is that all of this stuff is going to be released very, very quickly. And this will give a massive injection of greenhouse gases into the atmosphere over a very, very short time period. And that's going to be extremely dangerous. So I don't know if anybody really knows what the impact of that will be, but it's not going to be great. Well, the only kind of good news in this whole story is that the half-life of methane in the atmosphere is much shorter than CO2. So methane has a strong impact, but doesn't last so long. And the other possible thing that might be able to help us, especially for the methane clathrates, is that methane is not only produced by bacteria, but it's also consumed by other types of bacteria. So there are methanotrophs, which are also archaea, just like the methanogens, and they don't live on their own. They live in consortia with sulfur-reducing bacteria or maybe proteobacteria. So what is cool about this is this was a paper from a few years ago in Nature, and they found these consortia of A is for archaea, which are the methanotrophs, and these sulfur-reducing bacteria, which take the electrons from the methane and then they use sulfate as the electron acceptor. And the big question is, how does this electron transfer occur? When you have an electron transport chain within a bacterium, they have proteins within the membrane. You've got the cytochromes, the coenzyme Q, all this stuff. So you have molecules within the same membrane and the electrons get transferred from one to the next. So how does this occur? Well, they saw that these are proteobacteria, they're gram-negatives, and they produce pili that go out and make contacts with the archaeobacteria, and these are like wires that are conducting the electrons for this electron transfer. So, you know, there is a chance that if you have release of methane from the methane clathrates, it's not going to be released into the atmosphere, but in fact it will be consumed by these consortia of methanotrophs and sulfur-reducing bacteria. So it will only be released as CO2, which will have less of an impact. Okay, so getting on to the nitrogen cycle. Now I've copied over this diagram. Yeah, once again it's from Prescott, Harley and Klein's microbiology textbook, and it's got a summary of all the different nitrogen transformations that exist. Now, in order to understand this, I'd like to ask you this question here, okay? Because you have to try and figure out what arrow represents what process. Okay, so have a look at the white arrows and tell me what they mean. Ah, come back. Back here. Whoa, okay. Okay, yeah. Is it okay? It's open, right? Yeah. Yes. Oh, yeah, sorry. Ah. I'm so sorry. Oh, it's not working. Okay, wait. Yeah, it was on my screen. It's like it started, but then I read sometimes the votes aren't coming in. And I don't know. I have to renew the window and then click again on start, and then it works. But thanks anyway. Yeah, if you weren't there, that was it. You'd be here for like another half hour, and nothing would have happened, right? How are we doing this? Yeah. Okay, good. Only one possible vote for everybody. Yeah, right. So 20 votes come in. Oh, that's interesting. Yeah, the white arrows. Yeah, very interesting answers coming in. Everybody voted? Who wants to vote? Yeah, pretty much. Okay, let's have a look. So here's what you say. So you're. Oh, don't do that. Don't do that. If I kind of reduce the size of this window, I make it smaller. Yeah, that's what you say. Okay, what you're telling me. So, okay, so, okay, I'm just gonna, so I think, yeah, the fermentation, That's wrong. Okay, fermentation, you don't have any inorganic electron donor or electron acceptor. You start out with, for example, glucose, an organic source of carbon, and then somewhere along the line, pyruvate is the electron acceptor. It comes from the original glucose. So there's no transformation of ammonium, nitrate at all in any fermentation process. So this can't be right, okay? And the same thing is true for number 4, because aerobic respiration, you have organic carbon as the electron donor and oxygen gas as the electron acceptor. So you don't have any nitrogen compounds at all in this process. So really the choice is between these two, okay? And we have a majority of people saying that this is anaerobic respiration. Just so we're all clear about this, the question is about the white arrows that are going this way, right? And that's actually the wrong answer. The correct answer is chemolithotrophy. So why is that the wrong answer? The wrong answer is the wrong answer because in anaerobic respiration, nitrogen compounds are the electron acceptor, right? So it's nitrate that's the electron acceptor. So anaerobic respiration, that's this arrow here, right? This one. This purple arrow is anaerobic respiration, and also this one, and also this one. So the white arrows, those are the nitrogen oxidizers that live by chemolithotrophy. And they've got the names of the bacteria on here to help you out, right? Nitrosomonas, Nitrobacter. They are the chemolithotrophs, nitrogen oxidizers. Okay, so I'm not really so surprised that there's a big mix-up here, because it's not obvious at all why the arrow goes in one direction sometimes and the other direction the other times, and then all the time I'm saying bacteria can generate energy from this. So this is the weird thing, okay? We've got arrows going in both directions, and here I am telling you that bacteria generate energy from this process. And how can they both be true? Okay? They are both possible because it depends on who is the electron donor and who is the electron acceptor. So this process, reduced nitrogen being oxidized, is only possible as a source of energy if you have molecular oxygen, the best electron acceptor. This process here, anaerobic respiration, works to produce energy because bacteria have organic carbon available. So organic carbon is the electron donor, nitrate is the electron acceptor. So that's why, you know, bacteria can live running these reactions both ways. Depends who's the electron acceptor in the other half reaction. Okay? So that's the white arrow and the purple arrows. The white ones are chemolithotrophy, and the purple arrows, that's anaerobic respiration. Okay, that's cool. And then these ones are assimilatory nitrate reduction, so nitrate to nitrite to ammonium, and then it's incorporated into amino acids. Also, this is nitrogen fixation, okay. Dissimilation, this is when you have bacteria that use amino acids as their main carbon source. So they will excrete ammonium as the waste product. And that only leaves these two weird arrows here, which is called, oh, actually, I should do my little things here, which are things I've been talking about. And that only leaves these weird arrows here, which is what's called the Anamox process. So this was only discovered 20 years ago. And here, bacteria can use ammonium as the electron donor and nitrite as the electron acceptor, and to combine it to nitrogen gas. So that's kind of weird. They you can find them in the ocean, and you can also find them in wastewater treatment plants. So there's a kind of interest in whether we can use Anamox bacteria to denitrify water as part of the water treatment process, because otherwise you have to use denitrifying bacteria, and yeah, as part of that you need molecular oxygen to do that. Okay, so right, so Anamox bacteria. Now we have 20 minutes to think about photosynthesis in bacteria, and I think that will be plenty of time for all of us, right? Okay, so now basically two molecular systems used in bacteria for photosynthesis. So eubacteria use bacteria chlorophyll, which is directly related to chlorophyll in green plants, whereas archaebacteria use bacteriorhodopsin. Bacteriorhodopsin is what? Well, it's an integral membrane protein with 7 transmembrane helices, and it basically works as a proton pump. So this is the cytoplasmic phase, and this is the periplasmic phase of the bacterial membrane. Light hits the chromophore here, and that allows transport of protons across the membrane. So you get a proton motive force out of this process, and those bacteria can use it to produce ATP. No electron donor. So Halobacterium, photosynthetic archaebacteria. Things are a little bit more complicated when you look at photosynthetic eubacteria. So they all have basically the same biochemistry in the molecule that's going to absorb the light to produce energy. But when you look at them, they're all over the place. The different groups of photosynthetic bacteria do not have any recent common ancestor. So that means that they cannot possibly have inherited their photosynthetic system from a common ancestor. So how come they're all, so Chloroflexi, they're way out there, Heliobacteria, they're gram-positive, all the other photosynthetic bacteria are gram-negative. So when you have this kind of like dispersion of a shared set of genes, this is telling you there's been a lot of lateral gene transfer. One bacterium developed photosynthesis, and then the genes for it got transferred to a lot of different bacterial groups. Okay, so apparently what people generally think is that in fact photosynthesis was first developed here by the cyanobacteria, and then different parts of the photosynthetic system, molecular system, were transferred to different groups. So cyanobacteria are the only ones which have 2 reaction centers. Okay, so they've got both photosystem 1 and photosystem 2, the same as in green plants, whereas all the other ones either have 1 or 2. Okay, so that's why cyanobacteria are the only type of photosynthetic bacteria that can perform oxygenic photosynthesis. All the others can't do that. Okay, so if they're not performing photosynthesis, completely in the same way as green plants, what are they getting out of it? Okay, why they bother doing this? Okay, so I'm just going to look at 2 examples of anoxic photosynthetic bacteria. So one of them are the green sulfur bacteria, Chlorobi. So they've got photosystem 1 only. They collect light by a kind of antenna complex outside the membrane called the chlorosome. And basically what is happening is that they're using hydrogen sulfide as the electron donor instead of water, which you find in cyanobacteria and green plants. So what's happening here is that the electrons from sulfide or thiosulfide can be transferred onto the cytochromes that transfer it up to here, and then light comes in and zaps the bacteria chlorophyll, and then the electron can be transferred onto NAD+. What is the advantage for this? Well, it means that you can generate NADH directly from the electrons that come from the sulfide, which is not possible for sulfur oxidizers when they are living as chemolithotrophs. And that means it's more efficient, because that means the proton motive force doesn't have to be used to produce NADH, it can just be used to produce ATP. So it's an advantage for these bacteria to use this system. Purple non-sulfur bacteria, so they have a type 2 reaction center, so Rhodobacter is an example. And basically what they're doing is they're just cycling the electrons. Okay, so it's once it's on the reaction center here, light comes in, zaps it up, and then it gets transferred down the electron transport chain. The useful thing here is ubiquinone. Okay, so you have the quinone cycle as part of this electron transport chain. So it's going to pick up protons on the cytoplasmic face and release them on the periplasmic face. So you generate a proton motive force without any electron donor or electron acceptor. So it's a totally different system from what we saw in the archaebacteria, but the end result and the purpose and the usefulness for the bacterium is exactly the same. No electron donor, but the photosynthesis allows you to generate a proton motive force out of nothing. And then they can produce ATP using that. Okay, so cyanobacteria, these are, okay, they perform oxygenic photosynthesis just the same way as green plants. So here, you know, the electron donor is water. Photosystem 2 operates first. Then the electron comes back down through the quinone, so you get proton motive force. Then it's transferred to photosystem 1, and finally you get NADPH. So you can produce ATP and NADH just from light and water. Okay, so that's oxygenic photosynthesis. So here are, actually I didn't put Heliobacter in there. So here are the main types of photosynthetic eubacteria. They either have type 1 or type 2 reaction center. And basically you can see that, okay, cyanobacteria, these are the ones that are just living by photosynthesis alone. For the other types of green and purple photosynthetic bacteria, the photosynthetic apparatus seems to have been grafted onto something else. Okay, so if you think about this lateral gene transfer idea, the cyanobacteria, this seems to be the group that originated the whole thing. And then parts of this photosynthetic apparatus, the genes for them have been transferred to other groups. But if that's going to happen, then these bacteria already had to be alive and living with some sort of non-photosynthetic metabolism. Okay? So what it looks like is that the green and the purple sulfur bacteria, they were basically chemolithotrophs to begin with. Okay, so they're basically using sulfur oxidation as their metabolism. And then they acquire the photosynthetic machinery, they become a little bit more efficient. And they have an advantage in some environments, okay? Especially in anaerobic environments when there's no oxygen available. Because sulfur oxidizers, well, you know, they need an electron acceptor, right? They need oxygen. So that was basically transfer of some of the photosynthetic apparatus to chemolithotrophs, whereas the non-sulfur photosynthetic bacteria, Chloroflexus or the purple non-sulfur bacteria, they seem to be living as chemoheterotrophs. Often, if you grow them without light and give them a carbon source, they grow pretty well. Okay, so this is, once again, they were probably before they acquired the genes for photosystem 2, then they were just chemoheterotrophs. Okay, so I think that's about wraps it up for me. So this is just a summary of how bacteria are producing their energy. So respiration. The electrons start on organic carbon. They're transferred onto NADH. Electron transport chain generates proton motive force, and this allows you to produce a lot of ATP. At the end, you need an electron acceptor, which can be molecular oxygen or something else, like nitrate. So you need to understand why this is useful, high yield of ATP, and why anaerobic respiration is not the same thing at all as fermentation. This one needs an electron acceptor, fermentation does not. Chemolithotrophs, how do they live? Basically, they start off with some electron transfer from an inorganic donor. This can allow them to generate proton motive force, which can be used to generate ATP. And by reverse electron transport, you can get NADH. And then this is used to fix organic carbon. So it all starts with the electron transport chain in chemolithotrophs. And the thing that's tricky here is to understand why certain donor-acceptor pairs work and some others don't. What we saw with the question, ammonium as the electron donor and sulfate as the electron acceptor will not work. Chemically, you can't get an energy from that. And finally, for the phototrophs, you know, lateral gene transfer and how the acquisition of photosynthesis helps green and purple photosynthetic bacteria become a little bit more efficient. Now finally, so microbe of the week, we'll do one this evening. So 7 p.m. I'll send out the question, okay?